Now, after the proteins are modified in the ER, after they're modified in the Golgi and secreted from the cell, they're now going to be mature proteins. I want to review for a moment some of the important modifications that have to happen either co-translationally or post-translationally in order for a protein to function properly in the cell. So, while proteins are folding, of course, they may form disulfide bonds between cysteine residues. The cysteine residues from different portions of the protein might form covalent linkages to make these disulfide bonds. There might be other covalent modifications as well, but let's talk specifically about these 5 modifications. The first we already mentioned is glycosylation. As proteins pass through the endoplasmic reticulum, they can get sugars added to their asparagine residues. This is an N linkage. As the proteins pass through the Golgi, they can get sugars added to hydroxyl groups of either serine or threonine residues. This is O-glycosylation. So glycosylation. Next is proteolysis. There are certain proteins that actually, as they're passing through the ER-Golgi system, are going to be cleaved. And cleavage of the proteins is necessary for them to be fully active and catalytic. Some examples are like insulin. When insulin's made, it's attached to a C-peptide, but it's not going to be functional insulin until that C-peptide is cleaved and removed. There's zymogenic precursors like trypsinogen, chymotrypsinogen, procarboxypeptidase, proelastase. These are zymogens that degrade the food that passes through our intestines, but they're inactive until they get completely modified. And what I mean by that is that part of them has to be cleaved off or proteolyzed in order for the enzymes to be active. So it's just another way to post-translationally modify a protein. Here's a big one for biochemistry. It's phosphorylation. Now, phosphorylation probably occurs mostly out in the cytoplasm. You shouldn't think of it too much occurring within the ER and the Golgi, although we did just talk about a specific mannose phosphorylation that occurs in the Golgi. Most proteins, other than lysosomal hydrolases, are going to be phosphorylated in the cytoplasm post-translationally. Now, the enzymes that catalyze this are protein kinases. Protein kinases are going to take a phosphate group, attach it to a protein. Sometimes this activates the protein, sometimes it inactivates the protein. It just depends on the protein, okay, and what pathway we're discussing. So phosphorylation is a crucial post-translational modification. Many of these proteins have clinical relevance, and the one that has the most clinical relevance is the next one. It's gamma carboxylation. Now, gamma carboxylation is a specific post-translational modification that happens to 6 proteins that we'll worry about. What are those 6 proteins? Well, they're the proteins that are involved in coagulation. Now, which proteins in coagulation? It's clotting factors 2, 7, 9, and 10. Okay? 2, 7, 9, and 10. Those are the important clotting factors that get gamma carboxylated. Now, there's 2 additional factors that get gamma carboxylated, and those are protein C and protein S. If you remember, clotting factors 2, 7, 9, and 10 are procoagulant clotting factors, whereas protein C and protein S, those are anticoagulant clotting factors. That has some importance later on. Now, what does gamma carboxylation do? If you see down here, what it's going to do is to produce calcium binding sites. There's a specific amino acid on the clotting factors that will bind to calcium. But it's an area that has 1 glutamic acid residue. Because calcium is a divalent cation, there needs to be 2 negative charges for calcium to associate once it gets out into the clotting area in the periphery. So, in order to get the 2 negative charges so that calcium can associate later on, there has to be an additional carboxylic acid group added, and that's the job of gamma carboxylation. Can you remember what drug targets this particular step? Well, it's going to be warfarin, right? Coumadin. How does Coumadin work? Well, gamma carboxylation absolutely requires what vitamin? It's the coagulation vitamin, vitamin K. So Coumadin is going to work by blocking vitamin K's ability to participate in this gamma carboxylation reaction. So when you review your vitamins, you'll understand that gamma carboxylation, vitamin K dependent, is needed as an important post-translational modification for clotting factors 2, 7, 9, 10, and protein C and protein S. Very, very important. Now, prenylation. Finally, prenylation is when a group of fatty acids, or one or more fatty acids, gets attached to a protein. Now, what ends up happening is these proteins, such as G proteins, receptors, ion channels, they have to be embedded in the plasma membrane. So the G proteins get synthesized on ribosomes. They go through the ER and the Golgi. The receptors, they get synthesized in the ribosome. They go through the ER and the Golgi. The ion channels, the same thing. Now, how do these proteins get embedded in a plasma membrane? The way they get embedded is through this process of prenylation. Now, what is prenylation? As I said, it's the addition of a farnesyl or a geranyl-geranyl lipid group. What are farnesyl and geranyl-geranyl? These are intermediates that come off the cholesterol biosynthesis pathway. So these fatty lipophilic groups can attach to proteins, and when they attach to proteins, it helps anchor them in their lipid membranes. So anytime you have a lipid membrane-associated protein, chances are it got prenylated. Addition of a farnesyl or geranyl-geranyl group, it's kind of fun to say, geranyl-geranyl group got attached to its protein structure. If ever you get a case and a protein's described that is a transmembrane or a membrane-bound protein, and that protein's no longer going to be located at the plasma membrane, and you're asked, what was a potential cause of this? A legitimate answer is a prenylation site was mutated. Okay, so that's the clinical tie-in and how this might be asked of you on the exam.